Okay, good morning and thanks for joining us for this morning's webinar. So just a quick overview of what we'll be talking about today in terms of mango twig tip dieback. So we'll just have a bit of an overview about the research that's been done, and then we'll move on to what we've learnt from the bits of research, and then we'll have a look at some of the next steps and how people can get involved, particularly growers. So just briefly going over this timeline, this shows all the work that's been done so far. So it starts off in 2018 on the left-hand side of the screen there. So the department received some infected samples in 2018. And then as you can see, going along the timeline, all the work that's been done and the individual studies. And what we'll do today is just kind of briefly go through some of these and just highlight some of the key points that's come from these, and then we'll talk about what's happening next. So, what have we learnt from these studies so far? Well, let's just start at the start in early 2018, when the first pathogen isolation study was done by our teams. And what this found, what the aim of this was to try and identify any causal agents. So what it found was 150 fungal and bacterial cultures were isolated out of 22 samples. And what it found was that there were no exotic mango pathogens. There were 33 known species of fungi which have been associated with dieback. This included the bacteria Pantoia and the Botryosphaeriaceae family. Tests were also done and found that the phytoplasmas and Phytophthora weren't present at that time. So building on from that study, another study was done in late 2018, 2019. And this was to try and drill down further into the bacteria and fungi that were initially found. So what was done here was, first we'll start off with the bacteria just on the left there. So the Pantoia that was isolated out was used in a pot trial to try and see if that could be the potential cause of mango twig tip dieback. So 3 cultures were used to isolate healthy mango seedlings. And then the symptoms are supposed to be observed in the healthy seedlings, and then they're meant to be isolated back out. This test showed that no twig tip symptoms were observed in inoculated plants from the bacteria Pantoia. And then moving on to the right-hand side, 9 fungal species were found. The fungal isolates were used to inoculate mango seedlings, much like the bacteria. And again, symptoms were not observed in infected plants. So the next step was to have a look at some different diagnostic methods. And the team did some DNA next generation sequencing studies to try and take it a bit of a step further from the initial isolation studies. And this kind of came up with some interesting findings. So on the left-hand side there, it identified through this sequencing tools around 1,300 bacterial species were present. And what's interesting here was 67% of them, of the bacteria, were unculturable through the traditional methods that we used in the previous studies. So again, it came up with some of the 2 bacterial candidates that were initially identified, which is the Pantoyas. And then moving on to the fungi, this type of diagnostics identified 5,900 species of fungi, which is quite incredible. And again, the interesting finding out of here was that 60% of the fungi were unculturable, again through those traditional methods. It also kind of came out with 2 fungal pathogens to further consider, the Calochotrium and the Calosporidium. And sorry if I may have pronounced those a bit wrong, but you'll just have to bear with me. So then we'll move on to what was done next. So there was a small fungicide trial that was done. The aim of this was to assess if any possible chemical controls could be used. So the fungicides that were tested included Aero, Scholar, Mancozeb, copper sprays, Amistar, Sportac, and Octave. So these are just the trade names. This study established that Aero and Octave were most effective in inhibiting symptoms, even like in very small doses. However, it's really important to note that these chemicals may require a variation to the registration of the product before it's used in field-based control of dieback. So as with any usage of chemicals, make sure you check the usage requirements and the label requirements before you consider using any of these things. And essentially this study needs to be taken a bit further and looked at in orchard trials to kind of confirm some of these results. So moving on from the fungicide study, next so this is now kind of skipping a bit to late 2022. So the department commenced a series of transmission studies. This was to understand the mechanism of transmission, and particularly to see if transmission occurs via the soil, and to test whether the pathogen could be transferred from infected material into healthy seedlings. So the picture on the right shows an infected larger tree in the middle there, and off to the side you can see a pot in the background, and then you can see a smaller seedling here closer to the picture on the right there. So the planted healthy seedling was put into the ground, and the one in the pot was lifted up from the soil by a paver, so there was no contact with the soil. And what this found was that there was no transmission observed when the infected scion, like the twig, was grafted onto a healthy rootstock. So this study established that the transmission occurred between the infected host plants and the seedlings on the ground, but it wasn't transferred through the soil. It was transferred from maybe from higher up down to the healthy seedlings. So this starts to beg the question about, is this a single cause, or is it a combination of factors? So far we've just had a look at individual organisms in an attempt to identify a single cause for twig tip dieback. However, the reality is it's likely a combination of factors. And this is because many organisms are found in mango orchards without causing disease. For example, some bacteria and fungi live on the plant without even affecting it, and some only cause problems when the plant is under a lot of stress. So it's really difficult to identify a single cause. It's also really important to note that environmental and management practices can contribute to additional tree stress. So it's a really interesting thing to think about in terms of a whole of plant stress rather than just one individual organism causing problems to a tree. And just another point is that dieback research is really complex. And this has been demonstrated through research in dieback in other commodities. For example, there is a lot of work that's been done in grapevines and in pastures in Queensland, where almost a decade of research has been done in order to understand the causes. So it's very complex and it's a very long-term thing that we need to be thinking about. So this brings in the million-dollar question. So how can we manage it? At this stage, there is no single management intervention. Rather, there's a combination of methods that can be used in order to reduce the viral load. So it's really important to consider managing trees for year-round health. This is because when trees are under high amounts of stress, they are more susceptible to both fungal and bacterial pathogens. So in orchard management, we really need to start thinking about and promoting whole of orchard management practices and try and avoid the practices that may be adding extra stress to some of these trees. In terms of registered spray options, like we kind of touched on earlier with the fungicide trial, the options are really limited because essentially it was still very unclear whether it's a bacteria or a fungi or a combination of things. So using chemical treatments is a bit of a hit and miss approach at this stage. So if growers would like to have a chat about it, definitely go and get in contact with us here at the department and also try and seek some further advice from local agronomists. There's certainly some of those guys and girls that are doing a lot of good work in that space. And also, it's really important that everyone involved shares information. This will give everyone a better chance to find effective management options. So everyone's really got to share information about what their thoughts are, and then it will give us a better chance of success. So what are the next steps? So from 2018 to 2023, the NTGs has put in a fair bit of investment into all of those smaller studies that we just kind of briefly touched on. And as you can see from the little infographic here on the right-hand side, the NTG's put in approximately $500,000 worth of in-kind support and has done a lot of work to try and culture out what the cause could be. But unfortunately, it's very difficult and it requires further research funding to progress with some of the work that's already been done. So there's definitely need for larger and longer term studies to be done over multiple years and over successive seasons to really try and work out what's going on. And the last point here, I'd like to kind of make sure that everyone's trying to collaborate with each other and work with industry bodies, agronomists and government to try and all put our resources together and put our heads together to try and work through this issue because it is complex and it's going to require a lot more research to go forward to solve it. So how can you get involved? So if you're a grower, we'd really like you to get involved. AMIA is leading quite a few things on this. There's an online survey that's been put out by AMIA in conjunction with quite a few people. So the purpose of this survey is to be able to really understand how big the problem is. So then we can seek further support for more research. So this survey is just a quick online survey. All the responses are de-identified, so no names will be put on it or anything. But if you're a grower and you're affected by mango twig tip dieback, please make sure you have your say. This will be really important to be able to try and seek more funding, because we can't do it without industry support. So please jump online. It only has about 10 questions. So yeah, I'm just really keen to get people involved in that. So on the right-hand side there's a whole bunch of contacts. If you'd like more information, you can contact any of these people on the screen there, because this is something that will require further input from a lot of people. So definitely reach out. And also next year, AMIA will be doing a lot of infield surveys. So again, if you're a grower, definitely reach out to Celine there at AMIA and really get involved with that. That'll be really helpful going forward. So just some acknowledgements. I would really like to extend my thanks to a lot of people who have put in a lot of hard work into the information presented today, and to the people that have provided their thoughts and suggestions for this presentation. So thanks very much. And again, if you've got any more questions, please reach out to anyone involved in some of this work. So thanks very much.